The overall goal of the following experiment is to create cell microenvironments to study biological questions relating to individual and group cell behaviors that take place at micro and nanoscale dimensions. This is achieved by first designing templates that ultimately will guide cell placement. Next, these designs are then translated into patterned surfaces via photolithography, which allows for the creation of plasma shielding molds via soft lithography. Finally, these shielding molds are arranged as a tripod in a petri dish to allow for selective exposure of the underlying surfaces to plasma, which will leave a cell-sensitive chemical pattern on the areas under each mold. Results can be obtained that show how a cell's behavior depends upon its microenvironment and its interaction with neighboring cells.
This method can help answer key questions about the effects of microenvironments on cell behavior, such as how do interactions amongst groups of cells affect higher-order phenomena, and how do subcellular nanoscale cues affect functions such as health, migration, and differentiation.
First, use superglue to attach a patterned slide to a stack of blank slides. Next, create a container of aluminum foil slightly larger than the stack of slides and large enough to hold 30 grams of polydimethylsiloxane, or PDMS. Put the stack of slides in the container, and then pour PDMS over the stack of slides to create an initial mold. Then degas PDMS by placing the container inside a vacuum chamber. Break any bubbles present in the PDMS by quickly removing and replacing a finger over the intake valve tubing several times to briefly allow air to flow into the chamber. The valve is closed for 5 minutes of uninterrupted degassing. Then, allow the PDMS to cure in the clean flow hood at room temperature for 1 to 2 days.